We'll turn our attention now to my absolute favorite structure external to the cell wall, and we can orient ourselves back on our cell model and take a minute to look at what we call a flagellum. So a flagellum on this particular model cell is a polar structure, that is, it's at one end of the cell only. And sadly, when building this model, they didn't do a great job because they didn't make this movable. It would be nice if it was movable to show you that it can rotate either clockwise or counterclockwise, and those are the two motions, clockwise or counterclockwise. Those are the two motions available to the flagellum. So you get a sense for the structure, but you don't get a sense for the motion. But let's talk a bit more about it. So flagella obviously involved in motility. They are sometimes single or sometimes multiple, but they're always these sort of hair-like protein structures. They are hollow cored. So in the long filament region of the flagellum, it actually is a repeating protein polymer that that rolls around in a kind of helical way to leave a hollow core. So in fact, on this picture where we're really zoomed in to this particular cell wall, and a quick look at that will tell you whether it's gram-positive or gram-negative, right? So this flagellum emanating from the cell, and we can see that this is the filament. Further, this structure can be broken down into what's called the hook. And then lastly, this very basal structure that's embedded in the cell wall and membrane. And this is called, together, collectively, the basal body. So hook, filament, and basal body comprise the flagellum. Sorry, O. And further, the basal body is constructed of all of these different rings. And the rings are named the L ring. So this top one there is the L ring. The middle one here is the P ring. And bottom down here are the, this one is called the MS ring. And this is called the C ring. They used to call these the S and M ring, and I think they got tired of me making fun of that and saying it's appropriate that it stabilizes a whip-like structure. No, I'm kidding. It had nothing to do with me. But nomenclature changes, right? So recognize these as sort of the structures that comprise the basal body. So it's interesting to note that next to flagella, there are some really lovely protein channels that enable the influx of the proton motive force, the PMF. And it's the PMF rushing in. So you've got a hydrogen ion gradient right in the periplasmic space of gram-negative cells that can respire. And it's the hydrogen ion gradient swooshing in to proteins that span the membrane that actually gives rise to the rotation of the flagellum. So as those protons rush in, you get either the clockwise or counterclockwise motion of the flagellum. So we might collectively say then that the PMF is the energy source that's used to spin the flagella. In either a clockwise or a counterclockwise motion. And they're fast. In fact, the bacterium Vibrio alginolyticus can actually spin, spell this right, can actually spin at 270 revolutions per second. This literally gives rise to a bacterium that can move at such a fast rate that if we relatively compared it to us, and we say, I was going to pick on James and say James is running across the ultimate frisbee field, and probably James, you're super fast, right? But if you think about the miles per hour that you could go, compared to this bacterium, you just pale in comparison. If you were as fast as Vibrio alginolyticus, you could actually run at 82 miles per hour. Right? Nobody could compare, not even Usain Bolt. So not only are flagella rapid, like rapidly moving, give rise to incredibly motile bacteria, but also sometimes they can serve as virulence factors. So we might say that they can be virulence factors. That literally means that they can contribute to the ability of a microorganism to cause disease. So I want to bring up our good friend Helicobacter pylori. I have an awesome stuffed microbe, one of my favorite stuffed microbes. And you can see that as I passed this around to my students one day, it acquired a gangsta headband. But in any case, Helicobacter pylori is, maybe it's a misunderstood gangster, because in fact it can cause peptic ulcers. We know that a part of what it does is contribute to the development or the disease presentation of peptic ulcers. And one of the ways that it does that is because it has what we call Lophotrichus flagella. So check out this mohawk, but in fact these are a tuft of flagella at one end of the cell, Lophotrichus. And these rotate, and when they do, they enable Helicobacter pylori to bore into the lining of the gut. Okay, so this leads to the symptoms of a peptic ulcer. However, as I said, misunderstood gangster, because in fact, if this is in the right balance in your body, it can prevent acid reflux. So it does good things too. It's just when it's in the wrong balance that it can present as disease. That being said, we should say a little bit more about that distribution of flagella, ranging from those that have a single polar flagellum, being monotrichous, versus those that have multiple flagella at one pole, being one at each pole being amphitrichous, or multiple flagella at one pole being a Lophotrichus. So different distribution, different names for the flagella. So here we see the monotrichous flagellum. It's a single polar flagellum placed at one pole on the cell. And I was trying to remember, if it was Graham or maybe Peter, who got the unknown number 140, and that particular unknown, we were trying to see if it smelled like grapes, kind of like artificial grape flavor. That's very kind of sweet flavor that it has, or don't eat it, but smell the scent or odor. And I think you disagreed with me that it didn't smell like that. When you compare it to other bacteria, you may better hone that sense of smell. But in fact, it is a good example. So unknown number 140 is a great example of something that has a polar flagellum, whereas Helicobacter pylori is a good example of something that has a Lophotrichus distribution of flagella. And then lastly, peritrichous all over the surface of the bacterial cell would be what we would see for E. coli if it's flagellated. Not all E. coli are motile, but if they are, they have peritrichous flagella. So now that we can see the distribution of flagella on the cell, let's talk about how the rotation, the propeller-like rotation of flagella, will allow the bacteria to move in certain ways. That is, we know that they can rotate either clockwise or counterclockwise, and with that rotation comes certain types of movement. A counterclockwise rotation is going to be associated with the cell sort of moving forward, if you will, in a run-like pattern. We call it a run. That is a just straightforward movement. So when the bacterial flagella move in a counterclockwise way, we see them running. So we say that's a run. When they move in the opposite way, that is when they move in a clockwise direction, they sort of stop and reorient. We call that a tumble. So that's our stop, reorientation, that is a tumble. So this is moving directions or changing directions. So unlike James, when he's playing Ultimate Frisbee, and he can use what we call spatial sensing, where he can stand in one place on the field and he can see where the Frisbee is, he can move directly to that spot using spatial sensing. Bacteria would do no such thing. They use temporal sensing. That is, they sense one location that they're currently in, and in order to sense another location, they have to move. So bacteria have to constantly move in order to sense their environment and to tell whether it's changing. So the path that bacteria would take would be much more confounded than that James would. It would be more like a run, a tumble, a run, a tumble, a run, a tumble, a run, a tumble, and through a very convoluted path that we call a biased random walk, they would get to where they were going. So they don't use that straightforward spatial sensing. The process by which bacteria are moving in response to a chemical gradient is called chemotaxis. So while James is moving in response to where the ultimate, or where the frisbee has moved, potentially, bacteria, they're often moving in response to food. Now, not to say that James doesn't do that too. Really, I know my skiers, Elise and Ben are out there thinking, sometimes I move in response to food. And that's true. Bacteria often are moving towards a chemical attractant, that is a nutrient, for example. And their motion is, as we said, very convoluted because they'll sense that there's a gradient only by continuing to move. That is, they have to move in order to tell if where they are now is different than where they were a few seconds ago. So bacteria move towards a chemical or environmental attractant and away from a chemical or environmental repellent using chemotaxis and temporal sensing. That is, comparing the environment that they were in moments ago to the environment they are now in. So the chemical attractant or repellent enacts this change by binding to a transmembrane or integral membrane protein called a chemoreceptor. Once that chemoreceptor is bound up, it sends an internal signal that leads to the rotation either in a clockwise or counterclockwise way. So chemoreceptors affect what the direction of rotation of the propeller-like flagella. So they control the movement of the flagella. In the absence of a chemical gradient, there are equal numbers of runs and tumbles. So what we see is right down here, when a bacterium is not moving towards some sort of nutrient, some sort of attractant, then it'll just move like this, very, very randomly. It'll move from one spot to another, to another, to another, with the hopes of sensing a difference between the spots, right? It has to keep moving in order to do that because it can only use temporal sensing. So if it gets to one spot, say down here, where it's finally like, whoa, I sense something, then what will happen is that the number of runs will, or that the length of runs will start to get longer, and the number of tumbles will start to get less. So in the absence of a gradient, the bacterium has equal numbers of runs and tumbles, and it moves randomly. But if we put it in the presence of a chemical attractant, suddenly it's going to sense that, and it's going to begin to elongate its runs, more lengthy runs and less tumbles, until finally it takes this convoluted but a path that will get to the chemical attractant. So as compared to the random movement, we can see a biased random walk where eventually it gets from one low concentration in the gradient to the higher concentration of nutrient using this sort of movement. So this, again, is very convoluted, but it gets the bacterium there. And it's a dang good thing that the bacteria are wicked fast because they have to take a very indirect route to get to wherever they're going. Certainly we know people like that too. Whenever they drive, they take the most convoluted paths. It takes a while for them to get there, but they get there. And certainly some of them drive fast enough that they might beat you anyway, right? So I'll give you a moment to jot that down. And maybe you're thinking of some questions that you might have regarding the movement of bacteria with regards to a chemical attractant. And maybe you're thinking, yeah, but they might move towards food, but don't they also move away from toxins or pollutants or waste? That is, as more and more byproducts of their metabolism accumulate, bacteria are going to try to move away from those byproducts, right? We do the same thing. The more we pollute areas on the earth, we move away from them and leave them for someone else to deal with. This is what bacteria do too, right? They'll move away from their own waste. It's really interesting to note that the relative sensitivity for an attractant is much greater than that for a repellent. In fact, there's double the number of chemoreceptors for attractants as there are for repellents. And the sensitivity for bacteria for attractants is much greater. That is, they will move with response to very, very low concentrations of chemical attractant, and they require much higher concentrations of repellent to move in response to those repellents. So the stronger force, and I guess this makes sense, right? The need to eat is stronger than the need to get away from repellents. Makes sense. The last thing that I want to talk about are also structures external to the cell. And I'm going to bring back up good old Herbert, or whatever we've named our model. And we're going to find that in addition to a flagellum, so showing this rotating flagella that doesn't rotate, there are also pili or fimbriae on the surface of this model cell. So these pili, notice they're shorter and they're thinner than flagella, but they have some of the same ultrastructure. That is, they still have a hollow core, and they're still made out of protein subunits. They're arranged helically in order to create a hollow core, right? The same as what we saw with flagella. So the ultrastructure has some similarities. In terms of function, pili and fimbriae will allow for adherence to certain surfaces. For example, those bacteria, many of those bacteria that do cause urinary tract infections, will have certain adhesins, specific proteins, on their pili that allow them to bind to glycans on the urinary tract that enables them to hold on even when urine is voided. So that's a very functional ability of these pili or fimbriae. So they allow for adherence to particular surfaces. They can also allow for movement. The kind of movement that they enable is a really unique, really unusual movement. It's a short sort of quivering motion. And bacteria like myxobacteria tend to do this. And it's interesting how like populations of cells will move by, let's say, the short twitching movement. And it's given a really rad name. It's actually called, they're actually termed wolf packs. Like when they're moving this way as a population, they're called wolf packs. Very cool. So your book often differentiates between pili and fimbriae. Those terms are used interchangeably, but sometimes pili is reserved for the epic and dangerous sex pilus, which is the one that enables the transfer of plasmids, for example, from a donor to a recipient cell, that enables conjugation. And we'll talk more about that later, but suffice it to say that sometimes the term pili is reserved for that particular structure that forms between a donor and a recipient cell in the process of conjugation. So that being said, we're going to end there. I hope you have a fantastic Friday, and don't do anything I wouldn't do this weekend.